Hello, everyone, and welcome. My name is Robert Williams, and in this final presentation, I'll be exploring the effect of the mutated MUC16 gene on the survival of melanoma patients. To quickly run through what I'm going to be talking about in this presentation, at first, I'm going to go over the background of the MUC16 gene throughout the body as a whole, and then I'll be talking about my chosen study from the cBioPortal. I will then look at my research hypothesis based on some of the evidence I've gathered from literature sources. Then I'll be going through the cBioPortal figures and data that I have collected, explaining those and how they either support and or do not support my hypothesis. And then I'll be discussing some of the limitations of the data, the analysis that went into it, and then I'll be looking at kind of a summary of the presentation as a whole, and then the future implications of what I've discovered. And then lastly will be references. So first I want to look at the background relationship between MUC16, the gene, and cancer. So what is MUC16? This gene encodes proteins which help to form barriers around the epithelial layer of organs and systems in the body. Mutations of this gene activate two common pathways in cancer development, the first being the PI3K AKT pathway, which helps to stimulate proliferation, and the second is that when MUC16 becomes mutated, it cannot fulfill its role as a regulator of the GLUT1 protein, which further allows proliferation through the glucose mechanism. This gene is one of the most commonly mutated genes throughout all of cancer, and most predominantly you'll see it in melanoma in this study. So the study that I chose to focus on from the cBioPortal is a cutaneous melanoma study from 2024. This study has about 470 patients, and the MUC16 count is about 66.3%, so just under 2/3 of all patients. The average diagnosis range, as you can see, is around 35 to 80. And interestingly, this is a very important piece of information, is that the sample types is that 78% is from metastatic samples, and then 22% is from the primary solid tumors. Cutaneous melanoma as a whole is one of the most common yet aggressive forms of melanoma, with around 100,000 new diagnoses each year. This cancer develops in the melanocytes, which are the areas in the skin that allow for pigment, in the epidermis, which is the top layer of the skin. However, these can metastasize to other areas of the body, commonly lymph nodes, brains, lungs, etc., which we'll delve into a little bit here. So now that we've looked at the study from the cBioPortal, it's important to look at some of the background literature regarding the MUC16 mutation and cancer. One of the most prominent things I was able to discover during my research is that there is a correlation between mutated MUC16 expression and this concept known as the tumor mutational burden, shortened to TMB. This tumor mutational burden describes the total number of mutations in the DNA of the cancer cells in the genome, and it indicates how well cancer cells will actually respond to immunotherapy and immune responses. So this is important because immunotherapy is better able to recognize cancer cells that have this high tumor mutational burden, which indicates that cells that have a high tumor mutational burden will actually respond better and often result in better prognosis for the patients. So researchers have found that cancer patients that have a MUC16 mutation actually have increased response rates to treatment due to that tumor mutational burden, as the MUC16 gene acts as a marker for the TMB. And lastly, an important and interesting notion I found was that some studies have found that MUC16 in combination with other mutated genes, such as TTN, which is the most common mutated gene in this study, further the enhancement of recognizing the tumor mutational burden and again, further the prognosis, the positive prognosis for a patient. So with the information I have found from the background literature, I was able to craft a hypothesis that I wanted to explore in the context of the cutaneous melanoma study. This hypothesis is that given the ability for the mutated MUC16 gene to act as a genetic marker for genome integrity, expression of this gene especially when coupled with other commonly mutated genes in melanoma, will actually reveal an increased patient survival uh, for those patients that are focused on in this study. So now we're going to go into uh, the study itself, draft some figures, and see if the hypothesis based on the literature actually has some merit. So the first piece of information that I wanted to focus on was what type of mutation the MUC16 gene acts as in a melanoma patient. As you can see from the top figure, the MUC16 mutated gene doesn't act as a driver towards cancer or have any oncogenic nature. It actually acts as a variant of uncertain significance, indicating that it doesn't really follow any specific pathway that promotes cancer in these patients. In contrast to this, I included a uh, image of the BRAF mutation, which does act as a driver, um, leading to proliferation in the protein tyrosine kinase. You can see that it acts as primarily a driver, um, with little mutations acting as a variant of uncertain significance. But just from this, I want to focus that the MUC16 is not actively driving cancer in these patients, and is just a passenger mutation. This plot shows the overall survival for patients that experience a mutated MUC16 gene versus those with the wild-type MUC16 gene. This encapsulates all patients present in the study, and you can see from the plot that the altered group, those that have the mutated MUC16 gene, actually experience a higher probability of survival at each survival month interval for almost the entirety of the duration of time. This can be further expressed by looking down at the survival plot summary, and at the bottom right, you can see that the median months overall for survival is over double that for the altered group than the unaltered group. This kind of gives a broad notion showing that those with the MUC16 gene, a mutated MUC16 gene, do express longer or higher probabilities of survival and longer survival overall. Similar to the previous slide, this slide once again shows how the mutated MUC16 gene can influence the overall survival for patients in this study. If you look at the figure on the left, you can see that patients that experience the mutated MUC16 gene have a higher percentage of living to deceased than those without the mutated MUC16 gene. In fact, the proportion of living to deceased almost swaps. And this indicates that those patients that do not experience a mutated MUC16 gene actually seem to have a higher percentage of death than percentage of living. This is relatively significant for the overall study, as it shows that the people with the mutated MUC16 gene have a better chance of survival, have a positive chance of survival, while those without a mutated MUC16 gene actually have a negative chance in comparison to death. Looking at the figure on the right, we can see once again the length of time for overall survival for these patients. Those with the mutated MUC16 gene have a longer median overall survival, as well as a longer range of survival as compared to those with the wild type MUC16 gene. This data once again just reinforces the idea that in the grand context of the study, patients that experience the mutated MUC16 gene do tend to show better responses of survival than those without. Now that I've kind of explored the relationship between overall survival status and mutations in the MUC16 gene in the grand context of the study, I would now like to hone in on certain clinical attributes of how the mutated MUC16 gene can affect survival of patients in this study. This slide focuses in on a metastatic site, lymph nodes NOS, described as not otherwise specified in terms of the location of the lymph node, and how mutations in the MUC16 gene and mutated genome actually impact how well patient survival status occurs. In the leftmost image, we can see that patients with the mutated MUC16 gene actually do show higher mutational count than those without the mutated MUC16 gene. This goes back to my topic of the tumor mutational burden and how those that experience a higher tumor mutational burden actually show a better response to treatment and prognosis for the patient. Moving to the right two figures, we can see a pretty stark difference in terms of how patients with their biopsy site being at the lymph node non-specified locations of the lymph node um, show the overall survival. In the left one, you see that patients with the mutated MUC16 gene, 54% show that they are living, with only 45% showing deceased. However, those without that mutated MUC16 gene, it jumps to about two-thirds in the other direction, where almost two-thirds are deceased and only one-third are living. This is a relatively drastic change, and it really shows how mutations of this gene can really impact survival for patients. Now, in my research of the data presented in the cBioPortal, not all findings show that exact same relationship. So as you can see, we have a similar mutation graph, mutation plot versus the type of biopsy. This time the biopsy is more samples of skin, areas more commonly associated with melanoma, not metastasis sites. These are the primary tumor sites. Um, and unlike the previous slide where you saw this drastic increase in survival for patients experiencing um, mutated MUC16 genes at these sites, we don't see the same relationship for the skin samples. So you can see in the two figures on the right that patients with the mutated MUC16 gene at the skin of the trunk, compared to wild type, the proportion of living to deceased is the same between the two. The MUC16 mutation doesn't really impact how well a patient prognosis will be for patients with this mutated gene. And then again, the skin, not otherwise specified locations, slight increase in the living percentage, but it's not significant enough to associate MUC16 with a positive prognosis. So interestingly enough, MUC16 mutations at the primary tumor site in these examples are not showing that increase in survival. However, at the metastases site I previously showed, we see a pretty drastic increase in survival for patients with this mutation. Contrast between the metastatic lymph node sites and the skin sites for the tumors, let me explore the overall relationship between the MUC16 mutations at primary versus metastatic sites. While certain areas of biopsy, again these lymph node sites, showed a really strong correlation between MUC16 mutations and the survival status of the patients, we're not seeing this relationship throughout all metastatic sites from the data set here. In reality, if we're looking at metastatic sites with both mutated MUC16 genes, which you can see in the figure on the left, and the wild-type MUC16 on the figure on the right, there isn't that much difference in terms of patient survival, and the proportions between survival and death between these two cohorts. So what this leads me to believe is that while certain biopsy sites show this improved or this prominent correlation between MUC16 mutations and the survival, this isn't experienced throughout all metastatic sites and/or primary sites of tumor development. So now I kind of want to look at some of the limitations I found in this study, especially compared to the literature which I explored earlier. So this figure right here shows the overall relationship between the mutation count and the survival status for patients with these MUC16 mutations. If you recall, one of the big factors that I explored earlier was this tumor mutational burden, which is supposed to indicate how well patients can uptake treatment, immunoresponses, and from that correlates to positive patient prognosis. Unfortunately, if you look at the living versus deceased versus the mutation count, there's not much evidence to support that higher mutation count correlates to a better prognosis for living. And in fact, the deceased have a lot wider range, and the medians are almost exactly the same. So in the grand scheme of this melanoma study, the tumor mutation count does not directly correlate to better prognosis for these patients. So this kind of nullifies the tumor mutational burden aspect in regard to the grand scheme of the data. In terms of specific clinical information, maybe if you focus in on certain biopsy sites and/or age cohorts, you might see different relationships. However, in the grand scheme, we're not seeing this relationship. Another limitation that I discovered was that I didn't witness the relationship between other mutated genes such as TTN and MUC16 further increasing the survival status in patients. Certain literature sources push this notion that other coupling these mutations together further showed enhancement of the tumor mutational burden and should correlate to better patient prognosis. However, looking at these figures, this shows all patients that have the MUC16 mutation, and then shows the patients that have and do not have mutated TTN mutations. They show almost the exact same proportion of survival to death in the left image, which doesn't really promote the idea that this mutation on top of the MUC16 mutation is really helping further survival. And then the figure on the right, the overall survival status, the median of the survival is actually less for those with the mutated MUC16 and TTN mutation than those that just experience the mutated MUC16 mutation. Um, Both these uh, charts kind of show that these mutations together aren't promoting further survival beyond at least what MUC16 was doing on its own. So a few other important limitations that I want to consider um, regarding my data analysis and the overall study is first that not all metastatic and primary tumor sites follow the same trend as the overall status. So if you recall, the metastatic and primary tumor sites didn't show much of a positive correlation between mutated MUC16 genes and survival rate. However, when we focused in on a specific example, the lymph nodes, which are a metastatic site, these did show a pretty prominent response to mutated MUC16 genes and how patients responded. Patients with the mutated MUC16 gene had a better survival rate, and those without actually had a much larger death rate. So we can't really treat all metastatic and primary tumor sites the same. It might be more effective to work on a site-by-site basis when analyzing these. Another limitation is that since 2/3 of patients had MUC16 mutations, there's a much larger data set to pull from than those without, and this may have skewed some of the figures and factors one way or the other, leading for some not as in-depth statistical analysis. Next, a big factor is that there is no indication of what immunotherapy these patients received and how this impacted their survival. This is important because a large factor of how the MUC16 mutation impacts survival is by acting as a genetic marker for immunoresponses. It indicates that a patient can, uh, the cancer cells are open to immune responses and can be attacked by immune responses. However, without any indication of the therapy that these patients underwent, we do not know if this influenced their survival. And then lastly, there was no consideration for various other clinical factors such as diagnosis, age, sex, and/or other illnesses, all of which could have an impact on how patient survival looks and how the mutated MUC16 gene would impact this. With all this analysis said and done, what are my final thoughts, and what do I think some future implications may be for cancer diagnosis and treatment? So I do believe that there is enough evidence to suggest that in certain conditions, MUC16 mutations may correlate to better prognosis and overall survival in patients. As we saw in the overall data set, patients that experienced mutated MUC16 genes had a higher probability of survival than those with the wild type gene. And at site-specific areas, such as the lymph nodes, we saw that patients with mutated MUC16 genes did have a better survival rate than those without mutated MUC16 genes. I do think it's important to note that looking at the data set overall, in terms of if you look at all the metastases sites and the primary tumor sites, you may not see any of these correlations. And this again furthers the idea that this may be site-specific. You may see it better at certain sites, both in the primary tumor sites, maybe different areas of the skin, versus metastasis sites. Maybe you'll see it better in lymph nodes, as we saw, than other areas that might metastasize, such as the liver, lungs, etc. So overall, I do think that there is enough data that supports that MUC16 mutations may be able to predict patient prognosis when specifically coupled with certain clinical factors, again, such as biopsy site. I'm not confident in the ability of MUC16 mutations to act as a genetic marker for this tumor mutational burden, and I did not see any strong correlations between MUC16 mutations, other mutations, and an increase in survival rate. So going towards the future, MUC16 may be able to indicate how a patient's prognosis will develop given certain clinical factors, site specificity, etc. However, I think there is more research to be done in terms of other clinical factors and uh, aspects such as tumor mutational burdens and other genetic mutations which can impact prognosis. And now here are my references for this presentation. If you would like, you can take a look. And now I would just like to say thank you to everyone listening. I know this is a bit long, but I hope that you learned something important about the MUC16 mutation and its potential impacts on cancer prognosis in patients. Thank you, and I hope you guys have a great holiday season.